The learning objectives of this lecture are to get an introduction to what a eukaryotic animal cell is and what are its main components, and towards the end we will be comparing it with a plant cell. So we have already learned about the structural organization in living systems. You have an organism. The organisms are made up of various systems like digestive system, respiratory system, circulatory system. These systems are further constituted by organs. For example, here you're looking at the digestive system, which is made up of intestines. Then the organs are made up of group of tissues. Tissues are group of cells working together to perform a definite function, and cells are made up of organelles. So today we will be studying about the structure of cells, what are the organelles present in them, and what are their functions. To get a general perspective, human bodies are made up of about 30 trillion cells. You can imagine how many cells we are made up of, and it's so important to know what is the shape, structure, and general function of these cells and their organelles. To study a cell, you need a very important equipment, which is a microscope. So a microscope is an equipment that magnifies smaller objects and helps you visualize them. And the images that you obtain from a microscope are called micrographs. Now let's look at the parts of a microscope. So this is a compound microscope. It has a combination of lens. One set of lens is here and one set of lenses here. The magnification of these two lenses get multiplied and you get an overall zoomed image. So these are the eyepiece lens or the ocular lens, which are towards the eye of the observer. You have a resolving nose pin. Then you have the arm from where you hold the microscope. You have objective lens, which are towards the object because you put your object here. Then you have coarse adjustment. This is the knob that you move up and down to bring your object into focus. This is the fine adjustment. When you have your object in focus, to make it more clear and sharp, you use the fine adjustment. This is the stage where you keep your object. This is the light source, and this is the power switch to turn the light on. This is the condenser with which you can change the amount of light that falls on your specimen. And this is the base of the microscope. Historically, several scientists have contributed to our understanding of what a cell is. For example, let me bring my pointer back. Anton van Leeuwenhoek. He was a Dutch lens maker who made a combination of lens and used it as a microscope to observe simple protists or sperms. Then we have Robert Hooke, who published his work in Micrographia, and he is the one who coined the term cell because he thought that the cell looked like small rooms of a jail. Then we have Schleiden and Schwann. These are the two scientists who gave the cell theory, and the cell theory says that all the living organisms are made up of cells. So cell is the basic unit of life. Let's have an overview of eukaryotic cell. Eukaryotic cell has outer membrane called the plasma membrane. Inside the plasma membrane, there is this fluid phase structure called the cytoplasm. Actually, it's not a structure, it's just the medium. In the cytoplasm, there are several organelles. For example, you have mitochondria, lysosome, then you have cytoskeleton. These are the combination of proteins which provide a structure to your cell. You have peroxisomes, you have centrosomes, and the most important one is the nucleus. You also have the membrane folds called the endoplasmic reticulum and a further folded membrane structure called the Golgi. Ribosomes again are very important structures in the cell, and we'll talk about them as we progress through this lecture. I want to point out this structure, these finger-like projections from the plasma membrane. These are called the microvilli. Microvilli are present on the cell surface to increase its surface area for absorption. You also have some appendages or some extensions from the cell body, which could be flagella or cilia, that help the cell for movement or creating a flow around itself. The first structure that we'll talk about is a plasma membrane or the cell membrane. So this is the membrane of cell. It is made up of phospholipids, the structure that you're seeing here, and embedded proteins. So these are the proteins which are embedded in plasma membrane. Plasma membrane creates a selective barrier. It only allows certain things to pass inside the cell and only certain things to leave the cell and get outside. If you zoom into this structure, you will see that it is made up of phospholipids. Phospholipids are one molecule of glycerol, two molecules of fatty acid, and a molecule of phosphate on top here. We also talked about microvilli, which are the extension of plasma membrane. So you can see here in the wall of intestinal lining, the cells usually have these projections called microvilli for increasing the surface area for absorption. Next is cytoplasm. If you talk about a cell, there are three main structures. One is the plasma membrane, other is the nucleus, and in between these two is the cytoplasm. Cytoplasm is the cytosol, that means it is a gel-like material inside the cell in which all the cytoskeletal and organelles are present. Its composition includes several biomolecules like glucose, polysaccharides, amino acids, nucleic acid, fatty acids, glycerol, and ions. Cytoplasm is the site of various metabolic activities including protein synthesis. As we have mentioned that cytoplasm has cytoskeleton, which is a network of proteins that provide shape and structure to a cell. This structure is dynamic, it keeps building and breaking, so there are long chains or long pipe-like structures of these proteins which provide a skeleton to the cell, but they are made and they break continuously. For example, what you're seeing here in the video are long fibers made of proteins like tubulins or actins. If they are made up of tubulins, they are called microtubules. They play a critical role in cell division because they make mitotic spindle, which we'll study in coming lectures. If they are made up of actin, they are called microfilaments, and they play an important role in muscle contraction. Next, you can have several kinds of appendages arising from the surface of plasma membrane. For example, flagella or cilia. Flagella is usually one or few in number. They are thicker as compared to cilia, which are numerous. They are shorter and hair-like. What you're seeing here in the video is a flagella helping a sperm cell to move around, and what you're seeing here are several numerous cilia which are helping the paramecium to create the flow of material around it and also to move from one place to another. Next we have nucleus. Nucleus is the most important structure or organelle in a cell. The nucleus houses all the DNA that is required for passing the genetic information to the next generation. It also gives direction for protein synthesis in the cell. Then we have nuclear envelope, which is just the envelope around the nucleus. The nuclear envelope, just like plasma membrane, is made up of phospholipid bilayer. The nuclear membrane has several pores that you're seeing here, and these pores help in movement of ions, molecules, and mRNA from inside the nucleus to outside or from outside to inside. Inside the nucleus is DNA, which is condensed in the form of chromatins. Chromatins make chromosomes at the time of cell division, and in human beings there are 46 chromosomes, or you can say 23 pair of chromosomes. Inside the nucleus there is another organelle called nucleolus. Nucleolus is the structure that synthesizes ribosomal RNA. Therefore, they contribute in formation of ribosomes. Next we have endoplasmic reticulum. Endoplasmic reticulum is the membrane fold-like structure which is almost in continuation with the nuclear envelope. The structures that you're seeing here, they are of two types, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Let's see what is the difference. So the rough endoplasmic reticulum have these small structures called ribosomes on top of them, which is why they appear rough. Rough endoplasmic reticulum is mainly associated with modification of proteins. Smooth endoplasmic reticulum appears smooth because it lacks ribosomes, and smooth endoplasmic reticulum is mainly associated with the synthesis of carbohydrates, lipids, and steroids, and they also detoxify several medications and poison and also store calcium. Now let's say this is the nucleus. From the nuclear membrane extends the endoplasmic reticulum, and next membranous fold like structure is called the Golgi apparatus. So from the endoplasmic reticulum, once the proteins have been modified, they eliminate as the vesicles, and these vesicles enter the Golgi for further sorting, tagging, packaging, and distribution of proteins and lipids. So these vesicles, which are arising from endoplasmic reticulum, then enter or fuse with the membrane of Golgi apparatus. Inside the Golgi apparatus, there is lipid and protein modification, packaging, tagging going on. And once that process is complete, we have budding of vesicles coming out of Golgi apparatus. These vesicles can either fuse further with plasma membrane and the secretions are eliminated outside, or these secretions can remain inside the cell in the form of lysosomes or peroxisomes. So the lysosome which arise from Golgi apparatus are called the garbage disposal system of the cell, because they break down proteins, sugars, lipids, nucleic acid, and burn out cell organelles. Therefore, they are also called the suicide bag because they remove the dead, worn out cells organelles. Now, the pH of lysosomes is usually acidic, and they allow the degradation of proteins. Lysosomes destroy several pathogens such as bacteria and viruses. Thus, they also act as the defense of a cell. Since we have mentioned that the vesicles which arise from Golgi apparatus can form lysosomes or peroxisomes. We have just talked about lysosomes. Now let's see what peroxisomes are. So peroxisomes are small round organelles which are enclosed by a single membrane. Peroxisomes are associated with oxidation reaction. So they oxidize various substrates. For example, they oxidize the fatty acids and amino acids at the time of their metabolism. The byproduct of these oxidation reactions is peroxides. But peroxides are very harmful for the cells. So peroxisomes keep these peroxides or hydrogen peroxide contained inside it, and inside these organelles is an enzyme called catalase. The catalase enzyme breaks down this harmful hydrogen peroxide into harmless water and oxygen. Therefore, it's an organelle associated with detoxification. Peroxisomes also detoxifies alcohol. Next organelle is centrosome. Centrosome house these centrioles, which are also called the microtubule organizing center. So what you're seeing, these spoke-like structures are the microtubules, which form the cytoskeleton that we saw in the previous slide. These microtubules also form the spindle, and they help in the process of cell division that we'll see in the upcoming lectures. Another very important structure in cell is the ribosome. The ribosome has two subunits, the large subunit and the small subunit. The ribosomes can either be present floating in the cytoplasm or can be attached to the endoplasmic reticulum. Ribosomes are present almost in all kinds of cells, even in the prokaryotic cells, although they are a bit smaller in the prokaryotic cells. They are present abundantly in the immature red blood cells, where they synthesize hemoglobin, and hemoglobin, you know, is the protein that carries oxygen throughout the body. Next is the mitochondria, a very important cell organelle. Mitochondria is called the powerhouse of cell because it is involved in production of ATP, and ATP is the energy source for the cell. Mitochondria is this structure. It is a double membrane structure. It has an outer membrane and an inner membrane. The inner membrane is folded into the structure called cristae, and the mitochondria also has its own mitochondrial DNA. The space that's inside the mitochondrial membrane is called the mitochondrial matrix. Because mitochondria is involved with the production of power or energy, a lot of energy is required for processes like muscle contraction, so the muscle cells tends to have a lot of mitochondria in them. Till now we have studied the structure and function of animal cell. Now let's look at the differences between animal cell and plant cells, and then we will study the organelles which are unique to plant cells. So animal cells have plasma membrane, but the plant cells have plasma membrane as well as the cell membrane. Plant cells also have a huge central vacuole. Plant cells also have chloroplast, the organelle involved in the process of photosynthesis. Plant cells lack centrosome, and they also lack lysosomes, although in some studies you might find that sometimes lysosomes are present in plant cells as well. Cell wall is the rigid covering on the outside of plasma membrane. It provides protection, structural support, and shape to the plant cell. Cell wall is present in plant cells, fungal, and some protist cells. The cell wall is composed of cellulose, which is a kind of a polysaccharide. Next organelle which is unique to plant cell is the chloroplast. Chloroplast is involved in photosynthesis. It's present in plants and algal cells. It converts carbon dioxide and water into glucose and oxygen in the presence of sunlight. Let's look at the structure of chloroplast. It's a double membrane structure. It has an outer membrane and an inner membrane. Inside the membrane, there is this matrix space called the stroma and a structure called thylakoid, which is nothing but the folds of membrane. The stack of thylakoids is called grana or singular granum. Chloroplasts contain a pigment called chlorophyll, which entraps the sun's energy in the process of photosynthesis and leads to the production of glucose. The next structure unique to plant cell is the central vacuole. The central vacuole regulates the water content. It maintains the homeostasis of water content in the plant cells. Now, this water content is important to provide shape or rigidity to the plant structure, and the pressure created by this water content is called the turgor pressure. You must have seen when there is a lot of heat or sun outside, the plants lose a lot of water and they become wilted like this. But when you provide them enough water again, they revive like this. So the fluid content in these vacuoles are important for keeping the structure in a revived form. This fluid is usually bitter, which is why it discourages the herbivore animal from eating the plants, and they store proteins which are essential for developing seeds in the cell.